Well, hello and welcome to Bio 212, Anatomy and Physiology 2 at Moore Community College. And we left off with erythropoiesis. We're going to move on to white blood cells in a moment. But I just want to run through one more time what erythropoiesis is. And that is something that's going to disrupt homeostasis. And in this case, again, it's oxygen delivery to the kidneys. In which case, now the kidneys will respond by releasing erythropoietin into your blood. And think about what this actually means. If you go back to the previous figure inside your textbook, you're going to see now you have more erythropoietin. And as long as each of these is available in sufficient concentration, then you're going to make more of these. Okay? Just another way to think about it. Okay, so let's move on to white blood cells. And as you saw from the development flow chart earlier, you could see that white blood cells have nuclei. They do not contain hemoglobin, meaning they're not carrying any of the gases that we want to think about, meaning carbon dioxide, oxygen, or nitric oxide. And they're either granular or agranular. And that's based on the types of staining that's used. Okay, so we don't talk about histology in this class all that much, but if you can imagine, if you stain them appropriately, you're going to be able to see characteristics that will help you identify each of these, right? And we group them as either granular leukocytes or agranular leukocytes, and the neutrophils, eosinophils, and basophils. The way I remember it is granular nebs, okay? And neutrophil, eosinophil, basophil, agranular leukocytes. That means I don't have to remember these guys down here if I can remember neb, neutrophil, eosinophil, and basophil. How do we know what they look like? Well, let's take a look. Be looking for these things. If you haven't already seen them inside lab, you'll be looking for them soon enough, where you can see different populations. And this is what we mean by granules, right? You can see these granules. These are membrane-bound structures with specific materials inside of them. So the eosinophil will have maybe some histaminase, basophils will have histamine, neutrophils will have hydrogen peroxide, some other forms of peroxides, and each of these are exocytosed onto the target, whatever that's going to be, right? So what I just taught you there is that these guys release histamine. Okay, so now you know that basophils release a molecule that changes perfusion into space. So you're going to increase histamine. Okay, HIST. Okay, and to shut this down, eosinophils release histaminase. Histase. A-S-E. Oops, it's supposed to be an S. Okay? So these guys are actually, for some part of their responsibilities, antagonistic to one another. They have other responsibilities. Eosinophils, they do all sorts of cool things inside your body. These neutrophils, they're pretty much running around. Remember, they make up 60 to 70%, or 65 to 70%, or 63.2, 0.57%, or whatever. Okay? They're the largest number of white blood cells inside of your body at any given draw of blood. Okay? And they look for other. And if they see other, notice how large they are. You could fit roughly two red blood cells across this. They're about 16 microns in space. Here you can put about 1.5. Here you can put about 1.5. Okay? And take a look at that monocyte. You can actually easily get 2.5 neutrophils across, red blood cells across the surface of this thing. So you have a pretty good idea of what they're doing. Remember, lymphocytes, these come in three types. They either have T cell, B cell, or natural killer cell. You can't tell. Look at that. It's just essentially all nucleus, a little sliver of membrane, but that could be either one of those. The only way you can tell is when you look at the numbers and types of receptors on the surface of these things. Monocytes, they're slightly larger than the neutrophil itself, and you're going to notice it has no granules. There are no granules there. There's always going to be somebody who says, but that's a granule. Now take a look at the nucleus. Now you can see a large sort of horseshoe band-like structure, and that's what will help you differentiate them. And that's what I mean by looking for characteristics. Well, what are they doing? Okay, let's get rid of the cartoons. Move on to the next slide. You can see here that, like I said, white blood cells, they have limited lifespans, but T cells and B cells, they can live for a long time inside your body. They usually live for a few days. What does that mean? It means if you're a neutrophil and you have an active infection, that could be a really short-lived neutrophil because it's going to go off and actually kill itself to go save you. Okay? Excuse me. Sorry about that. Okay. Leukocyte, ptosis, okay, is the normal protective response of invaders where you're going to increase the numbers of these as a result of anything that's actually happening inside of your body. Notice here, strenuous exercise. You feel a cold coming on, go out for a run, right? That's what I usually tell people to do. And what's actually happening there is you actually end up with larger numbers of white blood cells. So whatever that pathogen is, if you have a large enough number, you can clean out whatever that was actually causing trouble for you. Okay? Its general function, of course, is to combat invaders. And I don't mean space invaders. I mean invaders as in something that's trying to turn you into a meal. Okay? And they do it by phagocytosis. They try to ingest whatever it is that's causing the problem. You'll see why that's really important inside chapter 22. Or by immune responses, which are actually part of this entire process. For now, what we're going to do is we're going to switch how they actually get around the body. I mean, how do you actually get these white blood cells inside the circulatory system to actually travel to peripheral tissues? And we call this immigration. Okay? They can leave it. Okay? Older textbooks would call it diapetes. Okay? Diapedesis. Okay? And what you actually have here is there should be plasma around here. And what you can see is there's a series of recognition steps that the neutrophil knows there's something going on, so it makes on its surface the integrin, all right? So that's that green square-like structure, and the actual basement membrane is down here. They've removed that, but you can see this is an endothelial cell, so that's an epithelial cell, and it's now making selectin proteins. So it's a selectin on the endothelial cell, and these two interact, and that's actually how the neutrophil knows how to get out of your circulatory system. It knows where to go into, right? And one way to think about this, you have a cut on the surface of your hand. How do those neutrophils know where to go? Well, there's a series of signaling steps that will tell it where to go, okay? Beyond the focus of this class. It's really cool stuff, though. Notice what it's doing now. When this interaction occurs, the tight junctions that were holding these two cells together are temporarily released. And it squeezes through. It's not like this thing opens up a hole to allow fluid to move into that space. The cell itself acts as a plug to make sure fluid doesn't come in with it. Okay? It's going to be precise for the different types of white blood cells that are actually working there. So, now that you know actually how they get out of blood vessels, neutrophils and macrophages are active phagocytes, meaning they travel around the body. Okay, let's draw macrophage. So remember, monocyte becomes activated. Rah, 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 rah. Okay, put some eyes on this thing. Okay, rah, angry. And it's running around the body. Let's say this is a bacteria here. It's screaming, help me, help me. Okay, so help me. What this does, it literally ingests it, opens up its mouth wider, engulfs it, rips it apart, and this is called chemotaxis, which means there has to be some molecule that's being left behind to find all of those bacteria. They must respond quickly, okay, to stop the tissue damage that bacteria is actually creating. I mean, because remember, you're a meal at this point in time. Bacteria is looking at you as though I'll do everything I can to destroy whatever I can of you. And to do this, it will use, meaning the macrophage or the neutrophil will use lysozymes, oxidants, those are the peroxides I was telling you about before, and a series of proteins that are called defensins. Okay? They get there first. Neutrophils get there first. There's no doubt about that. But monocytes, when they finally show up, they arrive in larger numbers, and they actually do more, okay, to destroy everything that's going on. When we think about let's get rid of the cartoon here. We think about what those basophils and eosinophils are doing. Okay, basophils, remember a moment ago I said will release histamine. Yay, so let's make sure you pay attention to that first, okay? So that means that we're going to change perfusion into a site. Perfusion is the amount of blood that moves into that space. We do this by vasodilation, which means histamine increases vasodilation. And what it does is it increases or intensifies the inflammatory response. Your body needs inflammation at specific times, but it also needs to be able to shut it off as quickly as need be. So you're going to see you have very limited numbers of basophils, you have larger numbers of eosinophils, so if some amount of histamine has been released, there should be more histaminase to shut that off. Okay? It's not the only thing that basophils do, though. They have heparin, which stops clotting, as well as serotonin. But all of these, heparin and serotonin and histamine, they're all signaling molecules. So I don't know about you, but that probably means to me that basophils are doing something really interesting inside the body. So if eosinophils make this histaminase to shut this off, what else are they doing? Well, they actually ingest antigen-antibody complexes. And this is going to be really important. You have to remember this when we get to chapter 22. So this small number of cells is inside the body, and those antigen-antibody complexes mean something. Okay? So we'll think about what that means later. And then they're really cool against certain types of worms, right? So I'll develop this a little bit more inside class, but essentially you're a meal. That's all you are. And I know nobody likes to think of themselves that way, but if it's not the bacteria trying to get you, you went out for a walk at some point in time and your bare feet inside the grass and you cut your skin and now there's a worm inside of you. And that worm is crawling through your skin, literally through your skin and into your body, and eosinophils are identifying them. Okay? And they're like, hey, you don't belong here. And all sorts of hell breaks loose after that. Okay, let's get rid of those. Let's go to here. Now we're going to remind you that B cells destroying bacteria and inactivating their toxins. You get to how that actually happens in a little bit later on inside chapter 22. T cells attack viruses, fungus, transplanted cells, cancer cells, and some bacteria, meaning there's going to be a receptor for the cells, the unique cell for any one of these things. And my favorite cells inside the entire body. That's not true. My favorite cells, partly because of their name and partly what it took to figure out what these cells were doing, is amazing. Okay? Natural killer cells, they attack essentially anything that isn't other, or isn't self, and is self, but doesn't look like self. Okay? Let me explain. So, we can attack infectious microbes. These guys just go in, they're just like, you don't belong here, and they just silence them. Now imagine, if you will, we make proteins, but sometimes proteins don't fold properly, or they're not made properly, DNA to RNA to protein. Sometimes the proteins just, they don't want to behave. Well, your body's like, well, we have an internal censoring system for that. And they send that protein to the surface of the cell. And that surface of the cell expression causes the natural killer cells to come and kill those cells. Now, it says, your textbook says certain tumor cells. The purpose there, of course, is it's a mutation that's driving some types of cancer. This is actually, in part, how they were discovered. Well, it turns out that, many non-tumor cells fold proteins inappropriately, and lo and behold, natural killer cells go find them. And it's an internal system for preserving natural protein production inside of your cells. Okay? Finally, platelets. And these are the megakaryocyte that destroys itself to become fragments. And inside those fragments, we actually have materials that will allow us to form this thing that's called a platelet plug. And inside those granules, they are a series of hormones that are going to allow for that plug to form, okay? Allow for clots to happen in specific, unique locations inside the body, so you don't have systemic clotting and systemic platelet plug formation, because you can imagine, hopefully immediately, what the consequence of that would be. So let's take a look at it. So, if we had all the time in the world, I'd tell you to stare at these cartoons and figure out what's going on. Well, let's remember here, yellow, these are endothelial cells. They're lining a blood vessel, and you're going to notice something here. The endothelial cells are not touching. And a few moments ago, I said during immigration that the tight junctions were lost here, but that the cell itself, the white blood cell that was moving into the space, blocked any action or any fluid that could actually touch that. Well, here it's significant. Here, what I want you to see is we start our story with collagen fibers, which are in the basement membrane, and damaged endothelium. allowing or revealing that collagen to be seen. And that's what the platelets are looking for. Platelets see collagen, and they freak out. Look at what's happening to them here. They liberate ADP, adenosine diphosphate. They liberate serotonin. Where have we seen that before? Okay, and thromboxane A2. Well, thromboxane A2 is a prostaglandin, and look at what's happening. We change the shape of the platelets, so they become flattened. You start to form these little fibrin threads inside here, okay? And they're releasing these molecules to now change what's actually happening inside of this space, and they formed a plug. They formed a plug on top of that to make sure that none of the fluid inside here ends up in the space over here, which is kind of awesome. So we go from adhesion to release reaction to aggregation. Now, this is part of blood clotting. It's not the whole piece to it, because blood clotting itself is where we're actually going to have the platelet causing the formation of fibrin threads, where you then actually will end up trapping materials inside of this space. And what this means is we are going to take coagulation factors that are in the plasma of your blood. They will react with calcium in a stepwise mechanism. And that calcium, as well as those inactive to active enzymes, will cause, in association with platelets, will cause the formation of that fibrin thread to create a clot. So there are three parts to this. You have an extrinsic or an intrinsic pathway. You'll see what that means. And ultimately, we have to have some activation of this molecule that's called prothrombinase. Okay? And prothrombinase is going to take the soluble portion. I think I just did something wrong. I got the spinning wheel of death here. Okay? Yep, I did. We're not going to print anything there. Yay. Get out of there. Fantastic. Sorry about that. Okay? Prothrombinase is going to be converted into prothrombin. It converts prothrombin into thrombin, right? You'll see why that's significant in the next figure. Okay? And then thrombin converts the soluble form, fibrinogen. Remember, I pointed that out to you at the very beginning inside A. Okay? And we're going to make it insoluble. Okay? So we're going to make fibrin, and those are going to be the threads of a clot. And here's the pathway. We're going to go over this in a lot more detail. Well, no, let's just do it now. Okay? Extrinsic pathway. We've crushed the tissue, therefore release this molecule called tissue factor. Tissue factor, in the presence of calcium, will now allow for activated factor 10. Factor 10 in the presence of factor 5 will, with calcium, allow for the activity of prothrombinase. Simultaneously and or independently, we can have exposed collagen fibers, where now those thromboxane A2 molecules and damaged platelets will cause activated factor 12. With calcium, with platelet phospholipids, we have a positive feedback system here that will allow for activated factor 10 in the presence of calcium and factor 5 with the presence of calcium, so that we actually end up with prothrombinase. Again, you could see if you have both of these activated at the same time, we would get to prothrombinase much more quickly, but independently they can do it on their own. Once we have prothrombinase, we enter what's called the common pathway, where in the presence of calcium, prothrombinase, and prothrombin, we take this and we create thrombin. And thrombin, with calcium, will take fibrinogen and start to make loose fibrin threads. And factor 13 becomes activated as a result of the thrombin and calcium and becomes the strengthening factor that actually creates the more dense, now insoluble fibrin threads. Okay? So that's a quick run through. And there's a few things I want to point out, okay? And that's tissue factor, or thromboplastin, leaks into the blood from cells, okay? And that's what's actually going to start the formation of prothrombinase here for the extrinsic pathway. Here, whoops, where we go? Sorry about that. Here, it's more complex and it's slower, but the whole idea is that once the platelets start releasing phospholipid into the space, as well as serotonin and all the other molecules, you're also going to activate the prothrombinase here. When we get to that common pathway, that prothrombinase with calcium converts prothrombin to thrombin. You can see essentially what I'm mentally doing here, and I'm hoping you're starting to do the same thing, is you're just simply drawing arrows inside your mind, and you're saying, oh, okay, I need this with this and this and that, and now I can get that. Thrombin with calcium converts fibrinogen into the insoluble fibrin, okay? What's not being discussed here is you actually have to be able to break this up as well. So inside class, and definitely inside your reading, you're going to see as the clot is being formed, right, we're also, clot formation remains localized, we're also putting materials inside here that will allow for clot dissolution. So I'm just going to put dis here, okay? Because you can't have clots inside your body for any prolonged period of time, right? You actually have to get rid of them. So literally, while you're building the entire clot, you're putting in the tools to destroy the clot over time. Okay, so I just want to finish up a couple of things here. And the idea that we're going to do is, actually, I'm just going to stop and I'll just put another one together.